Okay, thank you very much. Can I just check that you can still see my slides okay? Excellent, thank you. So yes, it's very nice to be invited to give the talk. I've been enjoying ESMTB events for many years. I think my first conference was 2008 in Edinburgh, which gave me one of my favourite anecdotes of science conferences, was that at the conference meal, somebody made the brilliant decision to sit everybody down in alphabetical first name order, which in particular led to there being a table of 10 Johns, which was interesting and is actually a point I'm going to come back to at the end of the talk. But yeah, it's really nice to be invited. Yeah, Ludek gave a pretty good summary of my research interests. I'm interested in interactions between host and parasites and infectious disease dynamics at a range of scales. So I've done a bit of within host modeling, a fair bit of epidemiology, but mostly I'm interested in evolution of hosts and parasites and their coevolution, and sort of how ecology and the environment drive those evolutionary dynamics. So the first part of this talk, I wanted to give a pretty general introduction to what I do sort of practically, and in particular talking a bit about adaptive dynamics. So I can see on the list of participants that there are some people who are, the world experts and even inventors of adaptive dynamics. So some of that will be fairly straightforward for some of you, but I just wanted to get everybody kind of up to speed on what it is practically that goes into these models. And then after that, I'm going to go through 3 different research directions I've been looking at over the last few years. Okay, so to start with a bit of motivation. So how is it that I think about evolution? So we're going to do a thought experiment. So let's imagine we've got a host population. So I've got a population of stick people here. I really need to change these figures because people are an awful example to use for evolutionary ecology, but they're what I've got. So we've got a population of stick people. So we've got healthy, susceptible individuals who are blue, and then those infected by some disease who are red. And let's assume that these hosts are able to evolve some defense mechanism to disease that means that they don't get infected so much. So they could reduce their susceptibility to being infected. But that this comes at a cost, which is sort of natural to expect. Hosts only have finite resources, so the more you put into defense, the less you've got left over for something else, reproduction, say. So given it carries these costs, a question we can ask is, well, what circumstances might we expect higher or lower defense to evolve? So the example here from kind of the first real host evolution paper in this framework from Boots and Haraguchi. What if we vary the mortality caused by the disease? So this we would often call the virulence. How would that affect host evolution? So on the y-axis here is susceptibility. So a low value of here would mean hosts are resistant to disease. They're not being infected much, whereas a high value of susceptibility would mean not much defense. They get infected really easily. And I think most people would presume that you want higher defenses against diseases that are killing you more. These are clearly causing damage to individuals and to the population by having high mortality and killing them. So presumably these are the parasites you would expect to see hosts evolve higher defenses to. So that would mean on this plot we saw a decreasing trend, so that you've got high susceptibility when mortality is low, but as mortality increases, you reduce your susceptibility and become more resistant. I think that's what most people would presume, whereas in fact we see the complete opposite. As mortality increases, we see susceptibility to being infected going up. So you've got highest defense when mortality is low and lowest defense when mortality is high. So that seems unintuitive, and what's going on here? Well, let's sort of work our way through this. So take the case where mortality is very high. So if the disease is killing infected individuals very quickly, that means that at any point in time, there won't actually be many infected individuals left in the population because they've been killed. Well, if there are many infected individuals in the population, then a susceptible individual, so this person in the square in the middle here, is actually very unlikely to come into contact with the infection. And if they're not likely to come into contact with it, there isn't the selection pressure there to evolve high defenses, given that it comes at a cost. So it might be really bad when you do get it, but you're so unlikely to get it that there's just not the selection pressure there. Whereas if the mortality is a bit lower, this means that there's going to be more infected individuals in the population at any time point. This puts more selection pressure on to evolve defenses, and therefore we see this higher resistance emerging. So I really like this as an example because I think it really highlights how ecology is driving the evolutionary patterns. So of course, evolution is fundamentally a process of genetics, but I think it's really crucial that we see how ecology drives evolution and also how evolution then feedbacks to the ecology. It's very much a two-way feedback. And that's really the points to take from this talk, I'll tell you from the start, is that ecology matters. So as I said, I wanted to start by giving some broad introduction to how I actually go about modeling evolution. And this is under the framework of adaptive dynamics, which many people here will be familiar with. But if not, let me talk through some of the basic ideas into this. So to start with, we assume we've got a population which is all identical, that has been interacting according to its population dynamics, and that it's reached a steady state, most generally an equilibrium of those population dynamics. So this dark blue population here has been existing for a while and it's reached an equilibrium. Then there is a rare event where a mutant type appears. So that's this light blue individual that has a slightly different value in one or more traits. So continuing our example before, perhaps it has a higher defense to disease, and then perhaps as a trade-off, therefore, has a lower birth rate. And the question is, will this mutant grow or die out? And that depends on its fitness. So we can define its fitness as its growth rate whilst it's rare. If that fitness is negative, that mutant population is just going to fizzle out and we'll still be left with the resident population that we had before. Whereas if the mutant has positive fitness, it's going to grow, and by and large, it will then replace the previous resident population and become the new resident itself. And so we now have a new population of light blue individuals, which again go along interacting according to their population dynamics and reach an equilibrium again. And so if we think about this pattern repeating, where we have a population at equilibrium, a mutant appears, tries to invade. If its fitness is positive, it does. It becomes the new resident. Then a new mutant appears, tries to invade, has positive fitness, so it does. So we can gradually move through trait space with the value of this evolving trait gradually changing. So let's go through a practical example of this, a really simple example for a disease model. So this is the model used in that Boots and Haraguchi example. So we have two compartments, susceptible and infected. It says prey there. That's just a little prelude to what's coming in a few moments. Yeah, just susceptible and infected individuals. Everybody is born susceptible. Only susceptible individuals reproduce in this model, I'm assuming. So infected individuals are completely sterile. Susceptibles have some background mortality rate. And then there is transmission. So we use standard mass action, density-dependent transmission, at which point susceptibles become infected, where they stay until they die, either through background mortality or through this excess mortality caused by the disease, which we call the virulence. So it's a very straightforward model, this one. So there isn't recovery in here. There isn't reproduction from infected hosts in here. Both of those should be, really, to make this a more general model. I just wanted to stick with something simple for this introduction. Okay, so we write down the population dynamics. I do try my best not to put too many equations and maths in these talks. I much prefer pictures, but just to prove I really do some maths, I thought I'd stick a few in. So we've got an ODE for susceptibles. So the first term, can I get a pointer or something? No. So the first term there, A minus, so A is the birth rate, Q is reduction in birth due to competition, B is the mortality rate, and beta controls transmission. And then infected individuals, again, they arrive there by getting infected, and they can die both through background mortality B and the virulence alpha. So we assume the resident population has gone to its equilibrium, and then we say, okay, a mutant appears, which we call SM. This has a different transmission rate, so beta is now beta sub m. And there is this trade-off, so that lower transmission rates are associated with lower birth rates. So A is now a function of beta m. And the important thing here is because the mutant is rare, any interaction terms only depend on the resident population. So in the brackets after Q, so this is the competition, the mutant is only competing with the resident, S and I. It's not competing with itself because that effect is negligible. And similarly for the infection term, we've only got infection from the resident infected population. So from there, we can write out the invasion fitness. So this is just the exponential growth rate of that mutant. So because we didn't have recovery or infected reproduction in here, that's a really simple term in this case. When you do have those processes in, you've now got a structured population, and you've got that those in the infected compartment are directly contributing to fitness, either through individuals recovering and being able to reproduce again, or infected can reproduce. So there you have to think a bit more about your fitness. Sorry, let me just go back to that. Yep, so then we've got the fitness gradient, which is telling you which direction selection's taking you in. So this is this idea of moving step by step through the different trait values beta. And then we look for a point where that will stop. And that's going to be where the selection gradient equals 0. So we can see at the bottom here that for this nice simple model, we just care how the gradient of the trade-off compares to the infected density. And so you can see in particular that if we had some defined trade-off, if we do something to the system that changes the infected density, that's going to change where on the trade-off the singular strategy is. Now, just like any other dynamical system, when we're at a stopping point, we need to assess stability. Sorry, can I just check that everyone is still seeing the slides properly? Because I'm just seeing a big picture of me on my screen.
We can see it, yes.
Brilliant, thank you. So there are actually two types of stability we have to worry here. So the first comes from classic ESS, evolutionary stable strategy theory, which asks if you are at the singular strategy, is that point a fitness minimum or a fitness maximum? So can mutants either side of you invade or not? And this is given by a partial second derivative, which in this model ends up just being the curvature of our trade-off. We then have the more traditional mathematical definition of stability as if you start slightly away from the point, are you attracted in towards it or repelled away from it? So this is made of two second order partial derivatives. And this, in this case, is made up of both the curvature of the trade-off and how the infected density depends on our trait of interest. Okay, so that's sort of the framework that we're working in. We write down equations for the population dynamics, we think, what are the dynamics of a mutant? Use that to write down a formula for its fitness. Look at how the fitness gradient looks, so for which direction evolution is taking you in. Where are the stopping points, which we call singular strategies? And then what are the stability properties at those strategies? And these lead to a number of different outcomes. So these are just a few key ones. So the first, so this is just demonstrating with outputs from simulations. You can do this all mathematically, usually, but simulations just demonstrate it quite nicely. So the first case here would be a point that is both convergent stable and evolutionarily stable. So you're attracted to it, and once you're there, no one else can invade you, so you just stay there forever. So that would be a continuously stable strategy. The case in the middle is a case of evolutionary branching. So this is a singular strategy that you are attracted to, so it's convergent, but when you get there, it's evolutionarily unstable, and mutants both sides of you can invade. And assuming a few basic conditions are met, the population will divide into two, and you end up with two coexisting strategies. So this is a really interesting case of evolution itself generating diversity. And then the last case is where if you've got two or more things evolving, you can get co-evolutionary cycles, where in this case, both the host and the parasite are having this constantly varying, fluctuating investment. So this would be a singular point, which is a repeller, but it's a repeller that's happened through a Hopf bifurcation. Okay, so hopefully that's just sort of a bit of a practical guide to how it is I think about evolution and what I actually do when I sit down to do some research. So I wanted now to move on and just to give an overview of a few different research questions I've been looking at in recent years. So, I mean, all of these involve starting from the point of standard host-parasite interactions, such as I've just discussed, and adding in some sort of complexity to the environment. So the first of these is thinking about the impact of predators. So the models I've just shown you and the models that were produced for many years assume that there was a host being infected by a parasite, just completely in isolation. But of course, the world is much more complex than that. You know, we're used to diagrams like this from school of different food webs, where we've got all sorts of different interactions going on. We've got competition between species for shared resources. We might have multiple parasites, multiple hosts, and in particular, the question that I've been thinking about is there might also be predators of the host. So in this case, we would go back to our SI model, and I've added recovery back in here to make it a bit more general. But we also have a predator. And we assume that the predator can eat both susceptible and infected prey, and that it can selectively predate on one or the other. And there's reasonable arguments why you might go in either direction. So in some ways, the more intuitive case would be that infected prey are slower, so they're going to be easier to catch. So you might predate more in infected prey. But it might also be that if infected prey give off cues that they are very infected and would not be a good food resource, then perhaps predators might be more likely to eat susceptible prey. I think that there are arguments both ways. And for this work, I've assumed that predators don't get the infection if they eat infected prey. So there's obviously a lot of very interesting scenarios in the real world where the life cycle of a parasite involves going through both prey and predator, and we're not looking at that case here. So first thing to say is that you can get all sorts of different things happening. So this is varying the predation rate and the selective predation, so how likely predators are to focus on infected over susceptible prey. And just to go through some of the acronyms here, so FSD is cycles, so it stands for fluctuating selection dynamic. So it's just the biological term for cycles. The BR means branching. So there's a region here where both the host and the pathogen branch. So you started with one host and one parasite, and you end up with two hosts and two parasites just through the evolutionary process. We've got a big region where the host branches and the pathogen maximizes its investment. This white region at the bottom left where the predator goes extinct. So we've got all sorts of different things that can happen, but we can also say something about where they happen. So in particular, thinking about this cycling region, we can see it's kind of funnel-shaped. So it's got a big region up to the top left, and it becomes less and less likely as you move down and to the right. So up at the top left there, that's low predation rate and high selective predation. So the combination of those two things will mean that susceptible hosts aren't being eaten very much, and infected hosts are being eaten a lot. And that this is the ecological scenario that's most likely to encourage cycles in host defense and parasite infectivity. So I think that's quite an interesting conclusion to draw, that these are the situations where we'd be most likely to see fluctuations occur. There's also some interesting results around when extinction of either the predator or the parasite can occur. So sorry, these figures haven't blown up too well on the big screen, but hopefully they still look okay. So this is just a phase space of the host's susceptibility trait on the horizontal axis and the parasite's transmissibility trait on the vertical axis. The big blue region is a region where the predator would go extinct if we were ever there. And then the red region is where the parasite would go extinct if we were ever there. And then I've put on an example trajectory. So it starts towards the bottom right, and we sort of spiral in towards the singular strategy at the blue dot, which is a CSS for both host and parasite here. So you end up with middling investment by both. Okay, now all I'm going to do is change the initial condition. So I'm going to assume that hosts started more susceptible with this dashed line. And you can see in this case, what happens is that dashed line moves into the blue region. Once we hit the blue region, the predator goes extinct. Now, once the predator's gone extinct, we can sort of imagine the whole phase space turning blue at that point, because the predator can't just suddenly magically emerge back again. Once it's extinct, it's extinct. So once it's hit that boundary, the selection completely changes because there's no longer a predator there. And we can see that this, in fact, leads us to go to a different CSS, where, in particular, we seem to have lower transmissibility in the parasite. So the coevolutionary process has caused the predator to go extinct, and then this has led to evolution to a less infectious disease. Now, what's interesting, excuse me, is that not only can the predator go extinct, but so can the parasite. So an important thing to know is that in a standard host-parasite evolutionary model, you cannot get extinction of the parasite through evolution, assuming defense is costly. And that's because as you approach the point where the disease would go extinct, so close to R0=1, basically, you get a smaller and smaller infected population. So we can think back to this example we had right at the start. The infected population is getting very small, so there's less and less selection. And because there's a cost, there will never be selection to actually go do that last step. So you might go to very, very low densities, and perhaps in a real kind of stochastic situation, we might see the jump to extinction, but in a deterministic model, you wouldn't see it. However, when a predator is there, it can happen deterministically. So here, this is just some different parameter values. So we can see we've still got blue areas where the predator would go extinct, red where the parasite would go extinct, and we've got this quite elongated trajectory here up to the CSS at the blue point at the top. And again, just by changing the initial conditions, we can have a trajectory that crashes straight into this red region at the bottom, at which point you can't really plot the trajectory anymore because the pathogen's extinct, so it's not evolving, and you've just got the host evolving and there is no parasite there, so the host will just give up defense because there's no reason for it. But what I think is quite interesting here is how we've reached that point. So if you look at that dashed line, we've started with low transmissibility and low susceptibility. And what's happened is the host has evolved to make itself more susceptible. So the host has lowered its defenses so that it's more likely to be infected by the parasite. So the parasite then gets all excited. It's got some hosts that it can infect really easily, which it does. This lifts the density of infected hosts, and then that's good news for the predator, because the predator here is selectively eating infected individuals. And then the predator eats so many of them that, in fact, it manages to drive them completely to extinction. So this is a really sneaky move by the host. It's made itself more easily infected, and yet that's ultimately led to the extinction of the parasite. So again, I think this is a really nice example of how ecology can drive evolution, but that then itself drives ecology. The host makes itself more easy to infect, we get higher infection, we then get higher predation, and that leads to pathogen extinction, and we're in a completely different ecological scenario then. Okay, so that's one thing that I've been looking at in the last few years. So now, changing tack, so a different question now is, so when I talked about how we do adaptive dynamics, I said we generally assume that the resident population has gone to an equilibrium, and that's because that's far easier to deal with. And for the classic SI style models, that's what happens. We get spiraling into a nice endemic equilibrium. But there are lots of situations where instead we would get fluctuations. So going back, in fact, to the predator example, I didn't say, but there I'd assumed simple type 1 linear predation, so that the more prey there are, just the more predation there is. Much more realistic would be to do a type 2 saturating predation, so that predation doesn't keep on increasing with prey density because of handling times. And we know that if you do that, that leads to population cycles. If your parasite has a free living stage, we know that that can lead to cycles. And in the work that we did here, what we did was put in a seasonality term. So obviously most ecological populations are subject to seasonality, particularly in birth rate, perhaps also transmission rate. So if we do that, we need to rethink how we actually do the adaptive dynamics, because we don't have an equilibrium anymore to substitute into our fitnesses. So if you think about what happens when a mutant invades in a case like this, there's a big problem with trying to take a point estimate of the fitness, because we can look at that and say, well, broadly, the mutant is growing, so that suggests that the mutant's doing well. But if I was just to take a point estimate, half the time, I could choose a point where the mutant density was actually declining. And so I would get an incorrect output from my model. So what we're more interested in than the point estimate is the general trend. And in fact, what's quite a good guide is the envelope that this fluctuating function is stuck between. I mean, this is one of my favorite curve sketching examples to give to undergraduates, when you've got a fluctuating function contained within an envelope. So the mutant dynamics are made up of some periodic function, which we may or may not know, times some exponential. And the exponent in that exponential term are the Floquet exponents, and they will tell you whether that envelope is going up or going down. And so I set a previous PhD student, Charlotte Ferris, the challenge of coming up with a nice routine to do this, and she did a brilliant job and came up with a really nice numerical routine for how we can just go through one cycle and then calculate these Floquet exponents. So what does this actually tell us? So here we're looking at host evolution, evolving its susceptibility again. And what happens if we change the amplitude of the seasonality? So seasonality was in birth here. So by changing the amplitude, we're making the birth rate go bigger and bigger and smaller and smaller. And what we see is that initially there's not much change, but as the seasonality gets quite high, then we start to see that the host becomes more susceptible, or in other words, less resistant. There's also a couple of other interesting features here. Right over the right-hand end, we can see that there's bistability in the evolutionary process, that there are two CSSs separated by a repeller, one at really very high transmission, so hosts that have extremely low resistance, and one more intermediate. And we also see that we have two fundamentally different ecological scenarios here. So the white region is where there is an annual cycle, and then the grey region is where there is a 2-year cycle. So many of you may know the seasonality in infectious disease literature, that we go through period doubling bifurcations to chaos. So here we've got a 1-year cycle and a 2-year cycle. And so again, we see how not just ecology drives evolution, but evolution changes the ecology. So this is a simulation of an evolutionary trajectory of a host starting with very low transmission and then becoming more and more susceptible, so getting to high transmission. And initially, it's in this region of a 1-year cycle, but then during the evolutionary process, it crosses this period doubling bifurcation and switches to a 2-year cycle. And then as it continues to evolve, we see the cycles become more and more extreme. So here the host has caused evolution into a much more complex environment itself. It's gone from a fairly regular yearly repeat into this biannual pattern with really quite steep peaks and troughs. So that's just host evolution. Charlotte then went and started thinking about coevolution. So what happens if the parasite is co-evolving with the host? Things are a bit different here. So up to now, defense has been about a quantitative amount. How well defended are you? In this case, we're looking at, it's more of an all or nothing thing, where high defense means you're able to successfully defend yourself against more parasites, whereas high infectivity in the parasite means you can successfully infect more hosts. So again, those of you familiar with the host parasite literature, it's kind of like a gene for gene type assumption, or we've called range in the past. Hosts can evolve to have defense against a range of parasites to be able to infect a range of hosts. So again, we looked at changing the seasonality, and Charlotte's work suggested that we should see declines in both defense and infectivity as seasonality increases. So we spoke to some biologist colleagues about this, and they've been doing some similar experiments. So Charlotte, as a mathematician, gamely volunteered to go and do some experimental work. So she went across to work in the lab of Mike Brockhurst, who used to work here at Sheffield. He's now in Manchester. Does lots of really cool work with bacteria and phage. And we did basically the same thing. She evolved bacteria and phage and varied the and put in oscillating conditions in the amount of resources available. And so these plots here go from the left, where the resources were constant, over to the right, where the amplitude of those oscillations were increased. And basically what we saw was, rather than a strictly decreasing pattern, we've got these sort of hump shapes. There's clearly a peak in the middle, where the resistance in bacteria, which are the host, is highest, and the infectivity in the phage, which are the parasites, is highest. So that was different to what we predicted from the model, where we'd thought it should strictly decrease. So we had to think about what's going on here, and in particular, we looked at what the fluctuations in births look like. So this is the left-hand plot here. This is how the maximum and minimum births vary with seasonality, and this is just the standard assumption that as you increase seasonality, your peaks and your troughs go up and down evenly. But that wasn't the case, so we checked this experimentally, and it was more like the left-hand plot here. So the peaks do seem to increase in a straight line, but the troughs had a much more interesting behavior. As you started introducing these oscillations, it was initially really quite flat. It didn't make much difference. And it was only as the difference in oscillations got large, that you saw these really quite big decreases. And so we rejigged the model to reflect this, so that the bottom, the troughs of our fluctuating birth rate, were initially fairly flat and then suddenly decreased. And what we saw in the results of the model now matched what we'd seen in the experiments, that both defense and infection peak at intermediate levels of seasonality. So this was really nice. It was great. It's a great experience for Charlotte to go in a lab and do some experimental work. And it was really nice to kind of go on that journey of, okay, here's what the model predicts. Let's check it against the experiment. Okay, it's not quite right. Let's delve into what might be going on. We found this pattern to do with the growth rates. So we came up with what looked to be a more realistic birth rate function, and then that produced results that matched. So it was a really nice bit of work. Okay, final research question I want to talk about is spatial structure. So everything we've seen up to now has been mean field. So we just assume susceptibles and infecteds live in this environment and just bounce around like crazy, banging into each other and interacting like that. Now, of course, that's not how it happens in most ecological populations. We have spatial and social structures. And there are different ways that you can introduce this idea, but the approach that I've taken in the past is to think of the population existing on a lattice. So you fix all your individuals in space now, so they're no longer bouncing around at random, but they're fixed on a cell in the lattice with either one individual or completely empty in each cell. Now you can still imagine this in your head, but say that there's still mean field dynamics going on. So if you're the susceptible person in the middle, so the blue person right in the middle of this grid, you could still be infected by anyone in the population. You can make that. But you can also go to the other extreme and say that actually you only interact with your 4 near neighbours on your grid, up, down, left, right. So in the previous picture, that susceptible person in the middle can be infected by any of those red individuals with equal probability. Whereas in this case, it's only the 2 red individuals, north and south, that could infect them. Okay, so how do we actually go about modeling this? Well, you start still with equations to model what we call singlet densities, that is, how many susceptible individuals on the whole grid, how many infected individuals on the whole grid. But if you've got this local transmission of disease and local reproduction, you also need to know how many pairs of sites there are. You need to know how many cases there are of a susceptible individual that's next to an infected, because that's how transmission happens. So you then need to have a new set of ODEs for all of the pair equations. So susceptible next to infected, infected next to infected, susceptible next to empty, infected next to empty, and 2 empties. However, it doesn't stop there. This is an open system. And so to know about the pairs of sites, you need to know about the triples of sites. I've not got enough hands. I need a 3rd hand somehow to have susceptibles, infecteds, and susceptibles, and so on. So what we can do is apply a pair approximation, which basically says once we get to the level of pairs, that's enough information, and we cut everything else off and ignore it. So again, the practicalities of how you do this. If it's a nice simple model, you can solve those equations analytically, but it's not really worth it, to be honest. And certainly once you've got a more complex system, it becomes too hard. So you solve them all numerically. You derive the fitness in much the same way. You think about what happens with a mutant growing. And so that's what's written on the bottom of this slide here. We can see we've got reproduction, there's that mortality term B in the middle, and then transmission, and that both reproduction and transmission have global and local components. So the global components are mean field. Just depends on how many empty sites there are on the whole lattice, or how many infected there are on the whole lattice for transmission. But then we also have these local components, which have these Q terms in there. These are conditional probabilities. So for example, the first one there is that you have an empty neighbor given that you are a mutant susceptible. And then on the right-hand side there, the probability that you have an infected neighbor given that you are a mutant susceptible. Now, there is something I think quite philosophically interesting here, and this is something that I think particularly the adaptive dynamics experts amongst you might have something to say on it. It's something I've kind of been debating in my head and not had a lot of time to think about. So in the standard adaptive dynamics formulation, when we write down the fitness, there's this important assumption of the rare mutant, which means that it doesn't affect itself. So for example, in that first example, we had competition in there, but it was only the resident densities that affected the competition on the mutant. And for infection, it's only the resident infected density that causes infection. For this model, that's no longer true. It matters. So mutants can affect each other. If you've got a mutant host next to a mutant host, that appears in the fitness, not necessarily directly, but indirectly. And so I think there's something really interesting going on here in terms of the adaptive dynamics, that now we've got mutants affecting each other that we don't have in the mean field case. And it's something, yeah, I'd like to sit down and think about more at some point what that means. And yeah, if those of you who do think hard about adaptive dynamics might have some interesting insights there. Okay, so we derive the fitness. So we find an attractor either by a numerical routine, or you can draw pairwise invasion plots. So no adaptive dynamics talk is complete without a pip in it. So here is one for us all to see. And then because we've got the pair approximation here, it's a really good idea to run fully spatially explicit simulations to back up the results. So a little screenshot of what that might look like. So there, black would be empty space, white is susceptible hosts, and red is infected hosts. So we can set up a lattice in the computer and run it with a Gillespie algorithm and see that those match up. So I started doing some of this work nearly 10 years ago, which is quite depressing. So for example, here looking at the evolution of host resistance, as we vary the amount of global reproduction, and we can see that as global reproduction decreases, so as you move to the left, as things get more local, you end up with more resistant hosts. So this is due to a combination of things. So partly to do with the fact that if you've got high resistance and you're reproducing locally, you create a little island of high resistance that's quite good for your mutant population. And it's also driven by the costs. So if you are investing more in resistance, you're lowering your birth rate, but in a weird way, they might actually be good for you because you end up creating more local space on the lattice. So there's then less intense competition for those empty spaces. So when we first did this work, we identified these drivers, but we didn't quantify them. And then a few years later, Florence DeBar did a really nice piece of work where she was able to quantify them and found that actually it was the costs that were the key here. So it's actually the fact that if you invest in high resistance, this lowers your birth rate, but the kind of unintuitively, this is good for you because it reduces competition in your local area. Okay, so this is what I was doing some time ago. And then my one slide to shamelessly plug my sponsors. So just, I mean, literally weeks before lockdown in March 2020, I found that I'd been awarded a Fulbright scholarship to go and spend a sabbatical at UC Berkeley, working with a biology collaborator of mine. So I then spent the next 18 months just assuming it wasn't going to happen. And then suddenly in kind of May, June time, the stars aligned and we managed to go. So I spent last summer, or this summer just gone, at Berkeley, working with Mike Boots and in particular a couple of his PhD students. And we didn't plan to work on this actually, but something that sort of fell out of our mutual interests was looking more at evolution in space. So in particular, I was kind of interested by this idea of, well, what if spatial structure in itself is a defense mechanism? So we can think of it in terms of dispersal. So if you're a host and you can choose whether you're, or not choose, you can, through evolution, have either your offspring predominantly local to you or dispersed globally, what should you do? So if there are no costs, you can show that you should fully disperse. You should always just sling your offspring off at random into the environment. But if we assume dispersal is costly, then things change. So we've looked at a few things. So this is very much, I mean, in prep is a very strong word. We're still working our way through much of this. So one thing we found is that as you increase local transmission, hosts would be selected to increase local reproduction. So we can see initially there's not much of a trend here. The line starts fairly flat and then just at the end it tips up. So this is suggesting that if infection is mainly local, you would keep your offspring mostly local, which in some ways seems unintuitive, but I think it's probably a case of if you've got lots of local transmission, you're causing demographic turnover because of the virulence. And so again, actually, it's a case of creating empty spaces on the grid in your local region. And so then it's good to reproduce locally. Something that sort of backs that up is that as we looked at what happens as you change the virulence. And again, as you increase virulence, you select for more local reproduction. So I think this is the same idea. If you've got higher virulence, you're going to end up with more empty spaces local to you on the grid. Which was all great, but then, so right back at the start, I talked about how it was really important to put recovery into models. The other thing that I think people are becoming increasingly aware of is that it's really important to put infected reproduction into your models. So this model didn't have infected reproduction in. We then put it in and the trends completely switch. So now higher virulence suggests for lower local reproduction. And I'm not entirely clear why that is at the moment. So this is a very recent result. We found this in the week before I left Berkeley, and then I've been teaching solidly since I've been back. So I've not had time to think about it. It's another clear example of where infected reproduction has a really important role to play. Okay, so what are the takeaways from this talk? So, as I said right at the start, ecology matters. And I think in particular that we get these two-way feedbacks, that ecology drives evolution, but then evolution feeds back and drives the ecology. And I think it's really important to recognize that two-way feedback. I've given a few different examples showing how complex environments impact host parasite coevolution. They can lead to different qualitative outcomes, cycles, branching, stable strategies, as well as different quantitative outcomes. So when you get high defense and high infection or not. And there's still loads to do, loads to explore. Now, before I finish, I wanted to take a couple of minutes to talk about equality in science. So this is something I'm very passionate about. I talk to undergraduates about this all the time in my teaching, and I talk to colleagues about it whilst we're sat around. And it always feels like the research talks are somehow sacrosanct and we should just talk about the research. But I mean, equality comes into everything we do. There is no research without equality in science. And I mean, I gave that anecdote at the start about, this ESMTB conference where there was this table of 10 Johns. There was not a table of 10 black researchers at that conference. And that's something that should really worry us. I mean, no offense to the Johns at all. There are some fabulous John researchers. But the fact that there would be more Johns than black researchers at a conference is a big issue. And I mean, mathematical biology, I think, as a field within mathematics, does a better job than many other fields, but it's still not great. And I mean, some classic examples. So this is a fairly famous study where lab professors were sent CVs for lab managers. The CVs were identical except for the name. It was either a male sounding name or a female sounding name. And there was significantly better rankings given to the male applicants. And this was regardless of the gender of the professor. So female professors were just as likely to show these discrepancies, showing kind of how implicit bias is embedded in all of us. We see it in citation rates. So this was a study where they came up with a model which should say, on average, how many citations people should have. So the prediction was that basically both male and female authors should have about 25 citations. And then when they looked at the actual numbers, the number of citations for the male was 25, but female researchers, it was 20. So female researchers getting 20% fewer citations than they should have done. And it's in data from grant awards. So this is UKRI data, UK Research Institute data. It's publicly available, showing that PIs who are white have significantly higher award values than ethnic minorities, and similarly, male PIs getting significantly higher award values than females. I don't pretend to have answers to this. These are massive structural problems, but I think it's important that we're all aware of them, and it's something I'm committing myself to in research talks to really highlighting this and making sure it's at the front of all our minds. So thank you for giving me the time to do that. So just time to thank some people. So everybody in my group over the last 10 years or so that I've been a lecturer. So the first 4 there are PhD students who've since gone on to bigger and better things, and then the other 5 who are still here. So in particular for the work today, Jaspreet and Charlotte Ferris made big contributions to all of that. Collaborators, so the fluctuating work, Mike Brockhurst and Rosanna Wright helped Charlotte with the experiments and were really helpful in discussions. And then that work on spatial structure at the end, Mike Boots and his PhD student, Graham Northrup. And then funders, Leverhulme Trust for some of the predation work, Fulbright for that spatial work. And then the hill that I'll die on is that most mathematical research is funded through teaching. And it's something that we need to get across to various people, that this is how we fund our research. But yeah, that's it. Thank you very much. And I'm happy to take a few questions.